Welcome to your third essential genetics video. Today we'll be looking at polymerase chain reaction. And essentially what polymerase chain reaction is, it's a process of doing DNA replication outside of a cell in a lab or something. So if you need a reminder on what DNA replication is, because we pretty much won't be going into it that much in this video, I'd recommend checking out the previous video on DNA replication, which is the second video in this series. DNA replication, polymerase chain reaction can be split into 3 steps. You've got step 1, you've got which is called denaturing. The steps might be called differently to what I'm calling them here, what they're called in my country, rather than in your country. Annealing. And the final step, elongation. Apologies for the camera again. This is just going to be a brief overview of PCR. I'm not going to go into the exactly what machines, what equipment you'll use. It's just a basic idea. This is really a high school sort of overview of the process. The reason PCR is polymerase chain reaction is because the products of one reaction, one DNA replication, themselves go through DNA replication, and then those products go through DNA replication, and so on, and the DNA molecules increase in number exponentially. They double at the end of each PCR cycle, each time you go through each of these steps once. So in step 1, well, before we get into step 1, I should list the ingredients you'll need in the reaction. So I don't have faith in my spelling abilities, so I'm not going to spell ingredients for you there. We need our target DNA strand, the DNA strand or sequence we're trying to make copies of. We're going to need primers. Now these aren't like the primers in DNA replication. These are made of DNA, not RNA. We're going to need DNTPs. That would be nucleotide, deoxynucleotide triphosphates. They're pretty much the nitrogenous bases on your DNA strand. They also provide the energy for polymerase to add them to the growing DNA strand. If you don't understand what the hell I just said, check out the previous video on DNA replication. Sounded quite snooty there, didn't it? Quite snooty way to say that. Now you're going to need polymerase, of course. This process involves high temperatures, so you can't use a normal polymerase, or like a polymerase you'd find in a human. You need to use a special kind of polymerase called Taq polymerase. Taq polymerase is quite resistant to high temperatures and actually works quite efficiently at high temperatures. The reason it's called Taq polymerase is because it's from an organism called Taq polymerase, the Firmus aquaticus, which thrive in hot temperatures. It's an extremophile, it's a thermophile. It's either a bacterium or an archaean or whatever. I can't really remember. And you're going to need a buffer to, I believe, make the pH of your this whole mixture of things to be the right temperature, the right pH for Taq polymerase to do its action. Okay, let's proceed with step 1, denaturing. Essentially what we're going to do is we're going to heat up, once we've put all our, we're going to heat up our target DNA sequence to about 95 degrees Celsius. So, top DNA to about 95 degrees Celsius. This is so we can break the hydrogen bonds between the nitrogenous bases in the DNA strand. So obviously this won't, in DNA replication in cells, usually helicase would break the bonds between the nitrogenous bases and unwind the DNA and stuff. But for this, we just heat it up. The next step is called annealing. In this step, we cool the DNA down to about 55 degrees Celsius. This is so our DNA primers can bind. Because obviously if hydrogen bonds can't form in DNA in the step 1, well, how come the primers are going to bind if the temperature is that high? So we just cool it down to 55 degrees Celsius. And that pretty much does it for that step. Step 3, our final step, is elongation. Now once the primers, it should be noted that with the primers, they're connected to the DNA strand in such a way that Taq polymerase can actually replicate both strands continuously, so there's no lagging strand and you don't have to deal with ligase sealing together Okazaki fragments. That's because we don't use one type of primer, we use two types of primer, sort of two sequences. So we just draw two DNA strands, just like so. With the 3' prime of one strand here, 5' here, etc. Here's one primer, and here's another. Sorry about that, I done the wrong ends on the DNA strands there. Of course, polymerase can only add DNTPs to the 3' end of the growing DNA strand. So this direction would be the 3' end of the primer. This would also be the 3' end of the primer. So these are two different sequences, let's say. Of course, Taq polymerase comes in, does its thing, extends the DNA strands continuously. Same with this one. But again, unlike in DNA replication in cells, this is continuous for both strands. And again, these primers are made of DNA, there's nothing special about them. I have drawn them in white. This is really just a force of habit, but these are the same as the other primers in the strand. And for this to happen, the DNA needs to be cooled down to about 72 degrees Celsius. Yeah, that's one cycle of PCR done now. That's it. And of course, we'd repeat the process again. We'd go back to denaturation, but with these two new strands now. We'd heat up the mixture back up to 95 degrees Celsius, break the hydrogen bonds apart, etc., etc. In the next cycle, this will become 4 new DNA molecules, then the next PCR cycle become 8 and 16, etc, etc. Now let's talk about the uses of PCR. At the end of each cycle, of course, the number of DNA molecules doubles. So after about 20 cycles of this, you're going to have about 1 million DNA molecules from one. So it can be used for things that need a lot of copies of a DNA sequence. Let's say you have a scenario where there's some notorious weirdo in your town breaking into apartments and sucking on people's toes. You're going to need, and they leave DNA at the scene. Let's say you've got very little DNA there, and you probably want to replicate that DNA so you have a lot more copies of it. It's pretty much for things like that. Or if you have fossil DNA from, like a very old fossil, you might want to make loads of copies of that. So if you lose one copy, you're not like screwed. Anyway, that's us. Next video we'll be talking about gene structure in prokaryotes. See you guys later.